Good morning class. Today we will study biomolecules. What do you mean by biomolecules? Biomolecules are the molecules which are present inside the living organisms. Can you name some biomolecules? Okay. Proteins, lipids, carbohydrates, vitamins, okay, and nucleic acids. What does nucleic acid involve? Your DNA and RNA. So here today we will be studying about the biomolecules which are present in our bodies. All cardinal features of life are summation of biochemical reactions. Where do these biochemical reactions take place? The biochemical reactions take place inside a body, and they are the reactions which take place amongst the biomolecules of our body. Now, building blocks of carbohydrates. The first biomolecule that we pick up is carbohydrate. Sugars and their derivatives. Glucose, sucrose, fructose that we use daily, that we intake daily, that are essential for our existence. The molecular formula are the multiples of CH2O, that is hydrates of carbon. Okay, carbohydrates. Molecules having up to 6 carbon atoms, they are known as monosaccharides. Now, what are the properties of monosaccharides? How do we classify them? What do we call them? First, we classify them based on the number of carbon atoms. If they have 3 carbon atoms, we call them as trioses. If they have 4 carbon atoms, we call them tetroses. 5 carbon atom, pentoses. 6 hexoses. Classifying them on another basis, that is on the basis of functional group, we have 2 types. That is aldoses, aldehydic group, ketoses having ketonic group. The combinations. If we say aldopentose, that means it has an aldehydic group and has 5 carbon. So, aldopentose. Similarly, if we give ketohexose, it has ketonic group and 6 carbon atoms. Now, glucose is aldohexose. What is the configuration which is generally present in our body? Carbohydrates are present in our body in D configuration. What are hemiacetals? They are monosaccharides. Hydroxyl and carbonyl group, they react to form hemiacetals. Internal hemiacetals are formed amongst carbohydrates which lead to formation of a ring structure in pentoses and hexoses. The 5-membered ring is known as furanose and the 6-membered ring is known as pyranose. There were scientists named Fischer and Haworth who gave their projections, who gave the structures of these rings and the linear forms of carbohydrates. Fischer gave the linear structure, the linear projection, and Haworth gave ring structure. What are anomeric carbon? The carbon carrying the aldehydic or the ketonic oxygen, which is C1 in case of glucose, that is carbon 1 in case of glucose, and carbon 2 in case of fructose. They are called anomeric because on formation of a ring structure, the carbon atom becomes asymmetric and it leads to formation of alpha and beta forms. That is why it is known as asymmetric or anomeric carbon. We have glycosidic bond in carbohydrates. What are glycosidic bonds? The OH group of one monosaccharide with OH group of another monosaccharide, when it combines to form a bond with the elimination of water, it is known as glycosidic bond, and the molecule formed by the combination of 2 monosaccharides is known as disaccharide. So, elimination of water is dehydration. When 3 sugars combine, that is 3 monosaccharides combine, it is known as trisaccharide. When 3 to 20 residues are there, it is known as oligosaccharide. And more than 20 to 50 residues are combining, then it is known as polysaccharide. What are the properties of sugars? The carbohydrates, reducing sugars. The sugars which are having free anomeric carbon, they are known as reducing sugars. There is a test for them. They reduce the alkaline solutions of copper salts to give yellow to red precipitate of cuprous oxide. So, Fehling and Benedict tests are performed, and these are the pathological lab tests for sugar in the urine and the blood. Sucrose is a non-reducing sugar. Now, we finish with the carbohydrate and take up amino acids. What are the building blocks of amino acids or the proteins? Amino acids together form proteins. We have 20 amino acids in our body. Two functional groups are there, amino group and carboxyl group, which are present in amino acids. The amino group attached to alpha carbon, that is the central carbon of amino acid. There is amino group in proline. Proline does not have amino group, it has amino group. And R group, which is present in the amino acid, is variable. Now, how do we classify amino acids? We have charged amino acids, that is having a positive or a negative charge on the R group. We have lysine, arginine and histidine, which are positively charged. There are two which are negatively charged, that is aspartic acid and glutamic acid. Where then the second part, we have polar amino acids with uncharged polar R group, that is they are polar but uncharged. Example, serine, asparagine, glutamine. The third category is hydrophobic. Hydrophobic R group is present here. Examples are glycine, isoleucine, proline, phenylalanine. The configuration of amino acids present in living organisms is generally L configuration. Now, what are the properties of amino acids? Variety of chemically reactive groups are present in amino acids like alpha amino, carboxylic group, phenolic group, sulfahydryl group. We have an anhydrin test to see the presence of amino acids. It is a reaction between alpha amino group of free amino acid and anhydrin. What actually happens? The anhydrin, which is an oxidizing agent, it reacts with amino acids, undergoing oxidative deamination, leads to formation of ammonia, carbon dioxide, aldehyde plus reduced anhydrin. Now the reduced anhydrin and ammonia react to form Ruhmann's purple. So appearance of a purplish color means amino acids are present there. But we have already discussed proline is an amino acid. It has amino group and not amino group. So proline gives yellow color with anhydrin test. So if we have yellow color, that means proline is there, and if we have Ruhmann's purple, that means the other amino acids are present there. What are the derivatives of amino acids? There are a few important derivatives of amino acids. They show us that without consumption of proteins, without the formation of proteins or amino acids in our body, it is very difficult for these processes to take place. The first example is neurotransmitters like GABA, gamma amino butyric acid. It is formed from glutamic acid. So it is a very important neurotransmitter. Our neurological signals will be affected if this amino acids fall short. The second is serotonin. It is formed by hydroxytryptamine from tryptophan. The hormones thyroxine, adrenaline from tyrosine. Plant growth factors like auxins, indole acetic acid from tryptophan. Mediators of allergic reactions, histamine from histidine. The vitamins and coenzymes. Coenzymes, the vitamins are important part of coenzyme. Without the vitamins, the coenzymes will not be able to function in a proper manner. The next are building blocks of lipids, the simple fatty acids, sphingosine, glycerol and cholesterol. Lipids are the constituents of our biological membranes. They are sparingly soluble in water. They are soluble in organic solvents like chloroform. The building blocks of larger lipids are long-chain hydrocarbons which are containing fatty acids. Examples, stearic acid, oleic acid, C18 amino alcohols which form sphingosine, glycerol, cholesterol. Steroids, they are derived from glycerol. Steroid hormone, estrogen, androgens, testosterone. Growth factors and immunosuppressive factors like cortisol. The properties of lipids, they are unsaturated fatty acids, they are saturated fatty acids. We have both the varieties present. The unsaturated ones have double or triple bonds. They add halogens